Welcome back from lunch. We hope you enjoyed a nutritious lunch. And now we'll start moving into our presentations for this afternoon. Our first presentation will present on Knowledge-Aided Assessment and Structured Application, also known as KASA, and how this will modernize drug product quality assessments. Our presenter, Dr. Andre Raw, contributed to the development of several important FDA initiatives, including the Guidance on Pharmaceutical Solid Polymorphism and Co-Crystals, Regulations on Listing of Polymorph Patents, Question-Based Review, and QBD Example for Generic Modified Release Products. He was instrumental in FDA's approval of generic versions of complex active ingredients, including Lovenox and Copaxone. He is currently involved in risk and quality informatics initiatives and is a principal architect of KASA. More recently, Dr. Rawl has been active in nitrosamine impurities. Within his tenure at FDA, he has been promoted to FDA agency expert and to chemistry division director. Currently, he is the associate director for science and communication in the Office of Lifecycle Drug Products in the Office of Pharmaceutical Quality. Our second presentation will discuss two ongoing initiatives to modernize regulatory submissions: revision of ICH M4Q and Pharmaceutical Quality Electronic Data Standards. Our presenter, Dr. Larisa Wu, is the Associate Director for Science and Communication in the Office of New Drug Products. Dr. Wu has been with the agency since 2011, starting her career as a chemistry reviewer. After OPQ was stood up in 2015, Dr. Wu was a special assistant in the OPQ immediate office and worked extensively on several FDA, CDER, and OPQ initiatives, including Knowledge-Aided Assessment and Structured Application, Concept of Operations, also known as CONOPS, for inspection of human drugs, process and facility integration, OPQ secondary assessment, and the BARDA FDA drug shortage program. Our third presentation is designed to help you quickly create a structured data file for a regulatory submission. Our presenter, Dr. Marlene Kim, is a registrar for FDA's Global Substance Registration System. She is a chemist specialized in chem informatics and computational toxicology. Marlene has experience supporting CDER's drug safety reviewers and quality assessors with quantitative structure-activity relationship, or QSAR, predictions and reads across assessments that were submitted by applicants. Please join me in welcoming our first presenter in this session, Dr. Raw.
Hello, my name is Andre Raw, and I want to thank the organizers of the SBIA DMF workshop GDUFA 3 Enhancements and Structured Submissions. In particular, in the context of structured assessment and submissions, I would like to give an overview on Knowledge-Aided Assessment and Structured Application, or KASA. But before I do this, it is important to define quality. A quality product of any kind consistently meets the expectations of the user. Drugs are no different. Patients expect safe and effective medicine with every dose they take. Pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. It is what gives patients confidence in their next dose of medicine. The Office of Pharmaceutical Quality, or OPQ for short, evaluates how a drug is formulated, how it is manufactured, and the facility used in the manufacturing process to ensure a safe and effective medication is being delivered to the intended population. OPQ also looks at formulation and manufacturing changes made after a drug is approved to ensure quality is maintained throughout the product's life cycle. Before I start talking about the KASA system, it is important to explain why we sought to develop in the first place. We did this because historically, our assessments have relied heavily upon freestyle narrative text consisting of unstructured information, summarization of application information, and copy and paste data. As a result, the assessment of product risk and evaluation of the applicant's mitigation approaches were dispersed in lengthy text, making it difficult to systematically locate and compare. The freestyle text resulted in inconsistency and encumbered our ability to share and manage our knowledge within FDA's repertoire of approved drug products and facilities. It also hindered our decision-making capabilities as assessors evaluated each application in relative isolation without fully assessing the wealth of information at FDA's disposal. We felt that this, in OPQ, that this was far from an ideal state and that we needed to modernize and move away from the unstructured narrative information in our reviews to structured data to enable a systematic approach for risk assessment powered by IT tools to best capture and manage knowledge. This resulted in our vision of developing the Knowledge-Aided Assessment and Structured Application, or KASA for short, a database platform for structured quality assessments and applications that supports knowledge management. Keeping this in mind, KASA was designed to capture and manage knowledge during the product life cycle, establish rules and algorithms for risk assessment, control, and communication for product, manufacturing, and facilities. It was designed to perform computer-aided analysis and provide a framework for a structured quality assessment. And during the past 6 years, subject matter experts at various levels developed, tested, implemented, and refined various homegrown structured assessment prototypes as a prelude to our present KASA system. Taking the learnings from these various prototypes and working with our colleagues in the Office of Business Informatics, Office of Strategic Programs, Office of Information and Management Technologies, all these efforts culminated in the launch of KASA into FDA's Nexus IT platform in the beginning of 2021. Following this release, termed KASA 3.0, in our IT roadmap, all incoming ANDAs for solid oral dosage forms were reviewed under this modernized and structured review system we term KASA. So what I would like to do is to go over the KASA platform and illustrate what this can achieve. This slide shows the actual landing page of the KASA system that assessors utilize once they log on to the system. As you can see, the KASA system is currently used by 3 review disciplines that evaluate these ANDAs. These include the drug product discipline that reviews the drug product design and its controls, the Office of Pharmaceutical Manufacturing Assessment that reviews the manufacturing process and facilities, and the biopharmaceutics discipline that reviews the dissolution as well as other aspects related to biopharmaceutics performance. Let me go more specifically into the drug product discipline. One of the various important facets of the drug product review is to capture or flag the inherent risk of the various drug product critical quality attributes such as dissolution, impurities, or assay associated with the drug product, and to capture how these risks are controlled or managed based upon drug product design or testing control strategies. As mentioned, one impetus of the KASA structured review is to ensure consistency and objectivity among our staff. This is achieved based upon objective risk ranking algorithms and structured descriptors for capturing risk control. First, the KASA has encoded algorithms that capture the inherent risk associated with the various CQAs present in the drug product. These algorithms, which use a failure mode, effect, and criticality analysis, or FMECA system encoded in the KASA IT software, objectively rate these inherent or initial CQA risks as low, medium, or high. This prospectively flags to the assessor the areas of high risk that need to be focused on, and conversely, the areas of lower risk that the assessor need not spend excessive time dwelling upon. Secondly, as equally important, the KASA captures the risk mitigation based either upon formulation design or control measurements using a drop-down with descriptors of structured knowledge for formulation design and control strategy measurement commonly used in pharmaceuticals. For example, if the CQA risk is flagged high for polymorph transformation of an amorphous form, one of those fundamental strategies in our drop-down is the addition of a stabilizing excipient to avert crystallization, among other strategies. In essence, these drop-downs, which are based upon our fundamental scientific understanding of pharmaceuticals, are descriptors of that structured knowledge for formulation, design, and control. And the importance of this is that this provides a consistent scientific dialect in our KASA assessments, which were previously lacking in our freestyle narratives. And by having these consistent descriptors, it not only ensures consistency of our assessments, but more importantly, it enables knowledge management, as I will allude to later. Remember, our traditional assessments were long narratives with cut and paste from the submission, which, as I mentioned, encumbered our ability to share and manage our risk information and knowledge. And here this slide depicts how the KASA enables a compact assessment. Once the assessor selects the fundamental formulation strategy to mitigate a CQA risk, then they have the option of explaining via a short narrative how that fundamental strategy was specifically applied to the application. For example, if they select the drop-down stating an excipient is used to stabilize the amorphous form, they would write a short narrative explaining what that excipient is, the API excipient ratio, and how that was systematically optimized in pharmaceutical development. And rather than cutting and pasting large sections of the pharmaceutical development report, they would simply link to the corresponding page in the ECTD submission. By adopting this strategy, this makes for a compact assessment where all the information is readily accessible in a highly structured format. Okay, now let's take one step back and put this all together. One of the reasons we developed KASA was to enable knowledge management, and this is nicely illustrated here. Based upon adopting the same inherent risk algorithm and drop-down of structured descriptors for risk control, we can now enable drug product risk analytics and can now systematically compare across a drug product type, e.g. an ROD and its corresponding various generics, how these risks are managed or controlled among different applicants. For example, first, we can see all the products are assigned the same risk for assay failure mode due to instability based upon our FMECA algorithm. Second, using the structured descriptors for risk control, we can now objectively see that while one manufacturer mitigated this risk by a de minimis approach of relying solely upon a stability testing program, others, to varying degrees, used formulation design features to mitigate this risk over and above testing. So by having this structured review, we can now run risk analytics reports in KASA to compare these relative product risks among different applicants depending on risk controls selected in these drop-downs. Thus, in the product life cycle and supplements or determining inspections at facilities, we can run these reports and use the information to allocate our resources to those products we believe are at highest risk. Similar to capturing risk control in a structured format, as I previously showed you, we can also capture the drug product control strategies such as specifications and the generalizable rationale for their control in drop-downs. By having this structured format similar to risk analytics, we can also run reports to compare these attributes across a product line. So we can determine if an incoming ANDA would have these attributes within the space of approved products, or conversely, if these attributes were outside our approved space. And as such, use KASA analytics to inform the risk of the incoming ANDA in question. So let me now go and illustrate the advantage of the structured KASA review in the manufacturing integrated assessment. You can see the KASA data structure for the manufacturing assessment is very similar to the drug product assessment, but rather than focus on product design and measurement control, it focuses on the various unit operations that may affect the various drug product CQAs. It similarly defines the inherent risk of the unit operations via algorithms and the corresponding control via drop-downs. But in this case, the drop-downs are a combination of associated process factors and facility factors. Similar to what I showed you for the drug product, we can now run reports based upon this structured data to discern the site's capability to manufacture these various dosage forms, as well as their prior history and experience with these unit operations. Such reports will reveal the risk of the site's demonstrated capability to run these unit operations, and with that, the degree of scrutiny required, as well as the need for an inspection. And finally, let me briefly go over the biopharmaceutics assessment in KASA. Biopharmaceutics is complex, and partly because of that, rather than invoking a paradigm of 3 risk levels, low, medium, high, for each CQA, in biopharmaceutics, they invoke 5 risk levels, from very low to very high for dissolution. And at one extreme, a very low, a simple standardized dissolution test would be sufficient to mitigate risk. And at the other extreme, a very high, in vivo studies to develop an IVIVC or IVIVR may be needed to support a patient-centric dissolution test. Rather than invoking risk algorithms to determine each of these risk levels, the biopharmaceutical assessment uses predefined decision trees that are encoded in the KASA software to guide each assessment as to where the product falls within these various risk levels. Similar to the drug product integrated manufacturing risk assessments, these structures not only provide for more objectivity of risk, but also enable KASA analytics related to the various aspects of biopharmaceutics. So, with all this effort toward development of KASA, we have made significant strides. I can say with confidence we are realizing our vision of knowledge management. This is evidenced by the fact that to date, KASA has 17 analytics reports that provide assessors with critical information for making informed decisions based upon KASA's structured knowledge of drug products and facilities. Just as important, we have taken significant steps towards solidifying the use of KASA among our assessors. And ever since Go Live in early 2021, as of early November, over 1,400 reviews across the 3 disciplines have been finalized for ANDAs for solid oral dosage forms. In sum, with the KASA 3.0 launch, we have made a significant step toward realizing our vision of structured review to ensure objectivity and knowledge management. But this is just one step of many in our roadmap as we move this paradigm to drug substance, to other dosage forms, INDs, NDAs, and BLAs. And next in this journey, we are on track to fully deploying the KASA framework to the drug substance evaluation early next year. One of the primary goals of the KASA for the drug substance is to use the structured assessment to enable informatics tools that can quickly identify risks in drug substance synthetic pathways that can potentially generate high-risk impurities, and thus a tool that can flag these higher-risk manufactured drug substances. In addition, the structured assessment will also ensure consistent standards for assessment of drug substance information across DMFs, ANDAs, and NDAs. And finally, the KASA will inform decision-making and increase efficiency of our assessment. This slide shows a pictorial of what the KASA drug substance interface will look like, including the various modules covering risk assessment, manufacturing, characterization, specifications, and stability. But specifically, in the drug substance manufacturing section, assessors will capture the synthetic pathway in a structured format, registering starting materials and intermediates used in the synthesis of the drug substance. Chemical structures of these starting materials and intermediates will be captured and, where needed, registered so they can be retrieved through the GSRS FDA database that is integrated into KASA. And although we can certainly do this manually, submitting these structures in an SD file format, which we strongly encourage you to do, will greatly simplify this process and make it more efficient for us. And several talks later in this session will cover this topic in much more detail. But ultimately, the rationale for doing all of this is to enable KASA drug substance analytics, where routes can be visualized, searched, and most importantly, analyzed with the goals of identifying reactions and or combinations that can generate potentially high-risk impurities. For example, this type of KASA analytics will enable OPQ to more effectively manage its repertoire of drug substances and target its resources to those manufacturing processes that have a higher risk of generating toxic impurities, such as, for example, nitrosamines. And to show our progress, this slide shows the actual drug substance assessment card in the Nexus system for ANDAs and corresponding reference DMFs that we intend to deploy in the first quarter of 2023. And with this future release, this will realize similar to what we have achieved for the drug product, manufacturing, and biopharmaceutics discipline, the concept of a structured review, and with that, our knowledge management of drug substance risk, including powerful tools to realize drug substance analytics. What is also more exciting is that this concept is not only applicable to drug substances manufactured in ANDAs or by reference to DMFs, but is also being extended to NDAs moving forward. As to KASA's future, we intend on expanding these concepts to other dosage forms, including liquid dosage forms, which encompasses a broad category of not only traditional injections for injections, liquid orals, but also topical creams, ointments, ophthalmics, nasal sprays, etc. And we also intend to extend this concept to other application types, including INDs, NDAs, BLAs, as well as the post-approval supplements. Actually, in relation to expanding KASA, which was initially conceived for ANDAs to now NDAs and BLAs, we even brought this to the OPQ Advisory Committee meeting early this month. And the committee unanimously voted to expand this concept to both new drugs and to biologics. This further validates our modernization efforts with KASA. In conclusion, the KASA system that we have developed enables the use of 21st century technology in our assessments and is driving innovation, for FDA. And this would not have been possible without all the efforts from countless OPQ colleagues, colleagues from our Office of Business Informatics staff, contractors, plus the steady support of our CDER leadership. And thank you very much for your attention.
Hello, my name is Larisa Wu. I'm the Associate Director for Science and Communication in the Office of New Drug Products, ONDP, at OPQ. And so far you heard from my colleague Andre Rowe about modernization of quality assessment at FDA through implementation of KASA. And next, it is my privilege to tell you about our efforts to modernize the regulatory submission, and in particular, to tell you about steps that we are taking to modernize the DMF submissions. So annually, OPQ reviews thousands of submissions that come to us in unstructured PDF format. We quickly recognize the need to modernize our quality regulatory assessment, and we are currently taking steps to transform our evaluation from narrative information to structured data and systematic approach for risk assessment, which is powered by the IT. And we do that in order to best capture and manage knowledge. This concept was envisioned in 2016, and it was discussed at the Pharmaceutical Science and Clinical Pharmacology Advisory Committee meetings happening in September 2018 and more recently in November 2022 as KASA. KASA stands for Knowledge Aided Assessment and Structured Application. It is a data-based platform for structured quality assessments and applications that support knowledge management. We already have a fundamental knowledge base of the products, manufacturing processes, and facilities. And in order to briefly explain KASA, one can imagine that as new information is submitted to FDA in applications, the assessors want to be able to review that information in conjunction with our existing knowledge and achieve knowledge management throughout the life cycle of the drug products that they evaluate. The system that we created, KASA, has the ability to capture and manage knowledge, incorporate rules and algorithms for risk assessment, and enable assessors to perform advanced analytics, which results in a comprehensive and risk-based structured assessment. We already implemented KASA for solid oral generics at FDA for 3 disciplines: drug product, manufacturing, and biopharmaceutics. And we're now getting ready to implement KASA for drug substance, which would be applicable to assessment of API information that is submitted in NDAs, ANDAs, and DMFs. And this release will happen in February 2023. To maximize the benefits of KASA, we need information and data that are well structured and organized. So let me give you a quick peek into the current KASA system. In the current system that we already implemented, as I mentioned, the assessor needs to manually enter unstructured information from the submission into the IT platform. And by doing this, the assessor structures the information to make it usable for KASA to automate and standardize the risk assessment and computer-aided analysis. But as you can imagine, this approach is not ideal, as it slows down the assessor who spends time on transferring data instead of focusing solely on the evaluation. And this really was an internal driver for us at FDA to initiate modernization of submission through a revised ICH-M4Q guideline, and eventually through having structured quality data that would enable the automation of some of the review functions. So based on this idea, we envision our KASA system as a 2-parts program. KA is the knowledge-aided assessment, an integrated set of tools and framework to aid regulatory assessment and knowledge management. And SA is the structured application, including content and organization of submission, as outlined by the ICH-M4Q guideline, and electronic data standards, as established by the FDA project called the Pharmaceutical Quality, Chemistry, Manufacturing, and Controls, or PQCMC. In the desired stage of KASA, an application would include structured, standardized data that would be able to auto-populate certain sections in the KASA system and therefore facilitate our assessment. So in the future, the KASA system would take advantage of the structured data in an application that would be compiled according to the revised ICH M4Q guideline and the new pharmaceutical electronic quality standards. So under the paradigm of a structured application, information such as specification or acceptance criteria that would be submitted as standardized data in the application will be extracted by automated tools and used to auto-populate sections in the KASA interface. And this would eliminate administrative tasks that we are currently performing and would allow the assessor to focus on assessment solely. So in an ideal world, our vision is to have both regulatory submission and regulatory assessment move to structured data format in order to enable efficient communication, knowledge management, and data analytics. We recognize this is a long process, but I think the first step, which is to transform the regulatory assessment, is already made through our FDA KASA initiative. And we're currently working on the second step, which is the regulatory submission transformation through our efforts to revise the ICH M4Q and to develop pharmaceutical quality electronic data standards. So in line with our KASA vision, we initiated the revision of Common Technical Document, or CTD, for the registration of pharmaceuticals for human use, or Quality M4Q R1 guideline. The main proposed revisions of the current CTD format are intended to optimize the quality sections in Module 2 and 3 of the application in order to capture drug product and manufacturing information in a way that would facilitate regulatory assessment and communication with the applicants. So what you will hear next is really the result of the work and ideas of a large ICH M4Q group that includes more than 30 members representing pharma organizations and regulatory agencies all around the world. So I'll start by providing a short background. The current CTD was first developed in 2002, but it did not become mandatory in select regions till 2016, and there are still regions where the CTD is not mandatory. The current format organizes the regulatory information into 5 modules with predetermined itemized sections, each section having various levels of granularity that vary significantly across applicants and submissions. In an eCTD submission, Module 3 contains all quality data that is pertaining to drug product and drug substance. And then this information is summarized in the Quality Overall Summary, or QOS, which appears in Module 2. The scope of Module 2 QOS is to give context and assist the reviewer in understanding and assessing the information presented in Module 3. We all know that currently this information is presented in PDFs, as I mentioned, and therefore cannot be easily searched, which makes lifecycle and knowledge management really challenging. And as I mentioned, this was our internal driver for submitting a proposal to revise the M4Q guideline. So back in May 2020, ICH endorsed the FDA proposal and agreed that the M4Q R1 guideline is due for revision in order to improve lifecycle efficiency, leverage digital technologies, and accelerate patient and consumer access to pharmaceuticals. The specific drivers of the M4Q R1 guideline revision were consolidated by the expert working group in the concept paper. You have the link here, and are outlined in this slide and the next one. First, as I mentioned, several ICH regions have not fully implemented ICH M4Q R1. The modernization of M4Q guideline is intended to support and clarify global understanding of the CTD, enable greater regulatory harmonization, and decrease redundancy. Second, the M4Q R2 guideline, so the revised one, should align with modern quality guidelines ICH Q8 to Q14 and other relevant ICH guidelines that have been developed since ICH M4Q R1 was issued. Third, the M4Q R2 guideline should provide guidance on the location of information supporting multi-component and/or complex products. Not lastly, the M4QR2 guideline should facilitate leveraging advances in digital tools, data management and standardization, and analytics to enhance efficiency and effectiveness of regulatory submissions and assessments, although, and it's important to mention here, the structured pharmaceutical quality submission is really beyond the scope of M4QR2 guideline. So with the revision of ICH M4Q, we intend to resolve a series of issues. The main issue to be resolved is expanding the scope of M4Q-R1 guideline. The current M4Q guideline does not serve well biological products, as I mentioned, so the revised M4Q-R2 guideline should apply to all pharmaceutical drug substances and drug products, both chemical and biological, that would require a marketing authorization. Second, we intend to establish the role of M4Q-R2 as the main source of the structure and location of regulatory quality information. More importantly, the guidelines should address diversity and requirements for quality information across ICH regions. Next, we want to resolve the organization of the product and manufacturing information in a suitable format for easy access, analysis, and knowledge management. Another issue to be resolved in the revised guideline is incorporating concepts and data expectations presented in ICH quality guidelines and aligning with current standards and guidelines. The M4QR2 should better enable the use of prior knowledge and ensure that the level of detail in the submission is commensurate with the risk of product quality. Next, the revised M4Q should better capture the pharmaceutical development and the proposed overall control strategy as the backbone of the revised M4Q structure. And this should address key elements of the proposed pharmaceutical product, including quality target product profile, or QTPP, manufacturing process, and overall control strategy. And finally, the revised M4Q will have an enhanced quality module 2 to facilitate the efficiency and effectiveness of regulatory submission and assessments. This quality module 2 may also incorporate key elements of ICH quality guidelines, including lifecycle management tools. Once the revised M4Q hopefully solves all these mentioned issues, we're hoping that it will also improve the submission and assessment efficiency, which eventually will result in accelerating patient access to pharmaceuticals. So the objectives of the M4Q-R2, as outlined by the concept paper and known as the 6 E's, are the following. First, encourage global convergence of science and risk-based regulatory approaches in the preparation of the dossiers. Second, explain and define the organization and positioning of information for modules 2 and 3. Third, enrich communication between regulators and applicants and enhance lifecycle and knowledge management. Fourth, embrace product and process innovation. Fifth, enable efficient use of digital tools for submission and assessment and prepare for the closely linked upcoming ICH guideline on structured pharmaceutical quality submission. And finally, elucidate regulatory expectations and support efficient assessment, decision-making, and actions. As you could tell, based on what I presented so far, the project really came a long way since May 2020 when the ICH endorsed the FDA M4QR2 proposal. And last year, we reached a few milestones, starting with the approval of the concept paper outline in April, and then the concept paper and the business plan were also approved by the ICH Management Committee on November 15, 2021. And at that time, the ICH Assembly also approved the formation of M4Q Expert Working Group. This group already met twice in person at the ICH Athens and Incheon meetings and is currently in step 1 with the goal of signing the technical document hopefully by the end of 2023. And all subsequent steps after step 1 are planned for 2023 to 2025, and that includes a step 4 adoption of the guidelines by the regulators. In 2025 and after, we are also planning the implementation step, including formation of a working group that would provide training as needed and would also monitor the implementation of the guideline. But keep in mind that all these dates are subject to change depending on how fast we build consensus on the technical document. So you heard so far about our efforts to achieve structured application through content and organization of submission via the revision of ICH M4Q guideline. A parallel effort, as I mentioned, is done at FDA towards establishing pharmaceutical quality electronic data standards. Specifically, I'm referring here to FDA PQCMC initiative. The current ECTD application is organized as submission components that contain unstructured narratives and data tables that are submitted in multiple PDFs. That significantly hinders the efficiency of data exchange, quality assessment, and lifecycle knowledge management. While KASA intends to change how submissions are evaluated by the FDA, the PQCMC initiative intent is to transform how industry presents the CMC data, the quality data in submissions, and is trying to harness a structured data approach. The PQCMC was initiated in 2014 at FDA that in order to establish content standards and electronic exchange standards for submitting quality or PQCMC data. And the objective of these initiatives are to first develop structured data elements for quality, and second, to design and develop data exchange standards that would make these quality data elements usable by various databases at FDA, including KASA. The initiative is intended to structure and standardize data elements in multiple phases. In first phase of the project, which was completed at the end of 2020, we covered elements including drug product, drug substance information, specification, batch formula, batch analysis, and stability. FDA piloted this concept with 7 industry members and evaluated the suitability of the standardized select quality data elements and terminologies. And based on the feedback that we collected, we continue to improve these data elements in conjunction with KASA. Phase 2 already started. We focus on in phase 2 on drug product and drug substance manufacturing, starting with manufacturing process data elements and terminologies. And obviously, work still needs to be done in that regard. But upon completion of these phases, the establishment of standardized pharmaceutical quality data elements and terminologies will provide opportunities for FDA and industry to transform quality submission data into usable electronic format that will improve the efficiency and quality of the drug product review process. Now that you have heard an overview of our ongoing efforts to modernize applications, let's see what we are specifically doing in terms of modernizing drug master file submissions, or DMFs. Currently, we are encouraging applicants or DMF holders to submit Form 3938 for DMFs, as well as structured data or SD files for chemical structures. And in the future, we may also need structured submissions of drug substance synthetic pathways. But you will hear more about all these efforts in the afternoon from my colleagues in the presentations that are following mine. So let's see how, starting with Form 3938 for DMFs. This standardized form is available for use with all DMF submissions, and it provides the agency with the contact and facility information in a structured format for easier use in KASA. It improves the efficiency and accuracy of information and saves the assessor time, as the corresponding KASA sections, as well as the letters for communication, can be pre-populated with the information in the form, which is pertaining to current contact information, facility information, or cross-reference DMFs. You can also help us improve our efficiency by submitting, as I mentioned, SD files for chemical structures. with your DMF. SD files are intended to minimize the manual work that the assessors are currently doing to draw and register structures in KASA. The SD files are text files that tell the computer how a chemical structure looks like. SD files are submitted, as I mentioned, by the DMF holders or the applicants. The original intent was to support the QSAR review, but we can also use these files to facilitate the registration of new chemical structures into KASA via Global Substance Registration System, or GSRS integration. FDA has been accepting SD files since August last year, and we're currently asking the DMF holders and applicants to voluntarily submit SD files. And you will hear more about how to submit SD files in the next presentation of this session, which is provided by my colleague Marlene Kim. So by submitting SD files with your application or with your DMF submission, you help us eliminate any transcription errors, you help us save time, and you help communication with other review function internally at FDA. We want to be able to use and search chemical structures for critical compounds such as starting materials, intermediates, final API, as well as impurities. So what we're doing right now is a lot of labor by recording chemical name, structure, all of these critical compounds in the synthesis. And we're doing so in order to register these chemicals and receive an associated identifier such as UNI number in GSRS that we can later use to retrieve these compounds. By submitting SD files for these chemicals, you really help us improve our assessment efficiency. But you may ask, what do we do at FDA with the SD files for chemical structures? We are using these SD files in KASA in order to capture the drug substance synthetic pathway in a structured format. And our intention is to capture the flow of the reaction steps, the synthetic inputs, outputs from each step, critical process controls, impurities that are carried over in the final drug substance, as well as any high-risk impurities that may result from the process, solvents, and reagents. Why we're doing that? It's because in a desired state, we would like to have drug substance synthetic pathways in KASA that are in a structured format that could be searched, visualized, and analyzed so we can easily mine information and inform our decision-making process. We want to be able to search and identify reactions and combination of reagents, starting materials, intermediates that can potentially generate high-risk impurities. And really, as I mentioned so many times, the ultimate goal is, again, to increase assessment efficiency and improve the drug substance knowledge management. So the take-home message here is that KASA for drug substance interface will be released in February 2023. It will be intended for assessment of drug substance information submitted in DMFs, ANDA, and NDAs. And this KASA interface, just like any other previous KASA releases, will present opportunities for knowledge management, consistency in decision-making, and improve drug substance assessment efficiency. Efforts are also ongoing at ICH level and FDA level to structure quality information submissions, as I mentioned. And you have to remember that the drug substance information can be currently submitted in a structured format in Form 3938 for DMFs and as SD files for chemical structures, which will eliminate potential transcription errors and further increase assessment efficiency. I would like to thank all of you for attention and also, most importantly, thank all the people that have been working on the projects that I presented. They have put in long hours, extremely hard work, and I know they are extremely dedicated people. Thank you for your attention, and I'm looking forward to the Q&A session.
Hi, my name is Marlene Kim. I'm a chemist and computational toxicologist. Today, I will go over the quick guide to creating an SD file for ECTD submissions. I must mention that any mention of commercial products, their sources, their use in connection with material reported herein is not to be construed as either an actual or implied endorsement of such products by the Department of Health and Human Services. This presentation reflects the views of the author and should not be construed to represent the Food and Drug Administration views or policies. Okay, so during this presentation, I will talk about how to create and submit a structured data file, also known as SD file. I will define what an SD file is. I'll go over the format. I will tell you what file locations you need to place it in the Electronic Common Technical Document, also known as ECTD. I will go over available public and commercially available software. I will also discuss special chemical structure scenarios that you may encounter while drafting your SD file, and I will also go over some additional support to help you. Now, before I begin, I'll give you a little bit of background. I would like to tell you what happens to the chemical structure at FDA when you submit your drug application through the ECTD. At FDA, the chemists go through the PDFs, and for the chemicals that they must review, they will manually redraw the chemical structures. This takes hours to days to redraw for each drug application. However, if the substances came in a structured format, such as an SD file with associated data fields, it will only take seconds to minutes to review the file or copy and paste chemicals for further analyses. Once the chemicals or substances are in a computer-readable format, FDA can register the substances. They can send it throughout FDA for consult requests. They can review it within the KASA platform. They can also perform computational analyses. Moving forward, in 2021, SD files became an acceptable file format in the ECTD. Industry may now submit SD files as a supplementary document with their applications. As of October 21, 2022, FDA received 65 SD files with an average of 30 structures per file. So what is an SD file? An SD file is a chemical data file format that can associate data with a chemical structure. The chemical structure uses the mole file format, which describes the atoms, bonds, connectivity, coordinates, and properties of the structure using strings of text. The file extension is .sdf, and it's also known as SDF. Now, on the left is an example of an SD file text contents. This is what you will see when you open an SDF file in Notepad, WordPad, or any text file reader. Then the computer will interpret the file and translate the string or the text into a graphical structure and table that you see on the right. On the right, you see the chemical structure and the data fields that were included. So unlike a mole file, which I mentioned earlier, a mole file will only include the chemical structure and does not include data. And unique to an SD file is that you can include more than one chemical structure and data. This makes it very beneficial for reviewing substances at FDA. Now that you know what an SD file is, I will go over the suggested file type and the file name. Please use the V2000 format. The other format that is available is V3000. The V2000 is preferred. One SD file containing more than one chemical structure will be expected. However, we understand that each application has its own needs, so please include what is needed for your application. For the file name, it is helpful if you write that it is an SD file, include the application type and number, and also include the name of the active pharmaceutical ingredient, the API. For example, the name can look like SDF underscore DMF underscore 012345 underscore loratadine dot SDF. Now in the SD file, if you add columns and data items to each chemical structure, it will make it easier for the reviewers to go over each chemical. Some helpful data items are such as the name of the chemical as referenced in the application number. Now, although you are submitting the file with the application, having the application number is important because you have to remember that reviewers and assessors review more than one drug application at a time. By having the application number linked to the chemical, it is easier to keep everything organized. Also include an ID, such as a unique identifier for cross-references. Include the role, such as the active ingredient, the process impurity, if it's an intermediate, degradant, metabolite, or starting material. If it has a UNI, please include it. The UNI is a unique ingredient identifier. You can obtain one from Precision FDA or from the public GSRS website that's hosted by NCATS. If it has a CAS, please include that. And if there are special notes that you would like to include about the substance that you cannot add in there in the structure, you can also add a column for notes. Now, these columns here on this page are just recommended, but we understand that you may not be able to get them all, and it's okay if some cells are blank. So here's an example of an SD file with the columns and data items and its associated data. Now it is helpful if you place the active ingredient in the first row or as the first record. So you have the structure, you have the ID, the name, the UNI, CAS, its role, and the application number. What types of chemicals should you include? So first and foremost, impurities of drug substances and drug products are the most important. Next, leachables exceeding the analytical evaluation threshold calculated from the safety concern threshold. Impurities evaluated by QSAR to support selection of a surrogate molecule or predict potential mutagenicity. And once you have created your file, we would like you to place it in Module 3. Module 3 has several sections which may contain information relating to drug substance or drug product. For drug substance, it goes in 3.2.S, and for drug products is 3.2P. Impurities evaluated by QSAR, if it's for a drug substance, it goes in 3.2.S.3.2. For drug products, it goes in 3.2.P5.5. Now, there is also a regional section in Module 3. Supported files placed here for any of the sections above should be properly hyperlinked. One SD file may be suitable when substances pertain to multiple sections. Non-clinical reports submitted in Module 4 may also include hyperlinks to SD files submitted in Module 3 to assist in cross-references. And there are also other sections, so you may also submit to other sections if needed. If it is needed for your submissions, you can also submit to 3.2 .S.6 or 3.2.P.7 for container closure system. If you're unsure, please reach out to your point of contact. Now, there are many software available that can help you create an SD file. There are commercial software such as ChemAx on Marvin, Microsoft Excel with the ChemDraw add-in from ChemOffice. There's also InStem LeadScope SDF editor, and also Molecular Operating Environment. And there are also publicly available and free software such as OpenBabel, Data Warrior, KNIME SDF Writer, RDKit, and also CDK. While you are creating your file, you may encounter special chemical structure scenarios such as salts, mixtures, 3D configurations, isotopes, or stereochemistry. In the event you encounter these, please use the SD file guide that's available on the internet, and I have the hyperlink available at the end of this presentation. For complex substances other than small molecules such as proteins, nucleic acids, or polymers, please contact FDA-SRS at fda.hhs.gov for a UNI. You can request a UNI at any time. So, an example of a special scenario that you may encounter is a substance with stoichiometry. Okay, in these events, it is best to include stoichiometry explicitly. For example, for magnesium chloride, we have 1 magnesium ion and 2 chloride ions. Okay, this should be explicit. For atorvastatin calcium trihydrate, you should include not just the atorvastatin, you should include the calcium and the 3 hydrate molecules. If there is an isotope, please specify the isotope, such as a urea C13. Please indicate which atom has the isotope. For ioben 1, I-131, okay, the iodine is mapped to isotope 131. In the event you need to include cyclic or ring substances, please use planar drawings for cyclic structures and ring systems. For example, for galactose, for D-galactose, this is the drawing on the left is the preferred drawing. On the right, we have the Hayworth projections, the chair projections, and Fisher projections. These are not preferred drawings because they can lead to confusion. For stereochemistry, please use wedge or dash bonds. Examples here are cholesterol and tetracycline. We simply use just wedges and dashes. Please do not submit 3D structures. Now for additional support, you may contact FDA-SRS at fda.hhs.gov. To request a UNI, you can ask about formatting SD files, and you may also inquire about how to draw complex structures. In most cases, we will provide you a UNI. If you have questions pertaining to DMFs, please email dmfogd@fda.hhs.gov. For questions related to CDER ECD submissions, contact ESUB, this is the ESUB team, at fda.hhs.gov. For questions related to CBER ECT D submissions, contact ESUBPrep, ESUBPREP at fda.hhs.gov. So we've come a long way from submitting PDFs to now we are moving towards structured data files that are machine readable. So we have standardized, so these SD files have standardized data fields that meet FDA's chem informatics needs. Errors associated with redrawing chemical structures are eliminated. And this also puts the responsibility on you to submit the right chemical structure. Submitting SD files also increases the accuracy, the quality review efficiency, and eliminates redundancy. Imagine once industry submits the structure, the reviewer only has to copy and paste that structure one time. Each reviewer does not have to draw it themselves. And this effort impacts drug substances and drug product reviews, QSAR analyses, and GSRS registration efforts. So we are asking you to submit a comprehensive SD file with your submissions. For additional resources, I have provided a link to a journal article called The Description of Several Chemical Structure File Formats Used by Computer Programs Developed at Molecular Design Limited. In this document, it will discuss what is a MOL file and what is an SD file in depth. For links to the FDA's SD file quick guide for DMF submissions, click here. You will click here. And for ANDAs, NDAs, BLAs, and all other types of submissions, you can find the hyperlink here. If you have questions about electronic common technical document, you may go to this webpage here. And if you have questions about study data, standard resources, that's also available at this link here. I would like to thank so many people who have made this effort come alive. So we have our SD file implementation team, Tyler Perrier, Barbara Scott, David Green, Naomi Krolak. We have our GSRS staff and collaborators, Frank Switzer, Larry Callahan, and Elaine Johansson, Tamika Mugoda, Marianne N. Kang, David Scanchi, Charles Robertson, Andrew Raw, Jeffrey Wu, and Extractables and Leachables Subcommittee. We also have our Data Standards Board, Jonathan Resnick, Heather Crandall, Helena Sviglin, Norman Schmuff, Jeff Florian, Marianne Slack, and Ray Wang. And there are so many others that have helped make this possible.
Thank you all for the great presentations. We'll now enter our first Q&A panel of the afternoon session. If you haven't had a chance to submit your questions in the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Joining us on the Q&A panel, we welcome Barbara Scott, Review Chemist in the Division of Lifecycle API, Office of New Drug Products in OPQ. Looks like we have some questions coming in right now, and the first group of questions will be directed to Dr. Andre Raw. And here's the first question for Dr. Raw. When will the KASA be implemented for DMF assessment?
Yes, hello. The KASA, as it was noted in the previous presentation, the KASA will be implemented, by March of 2023 for DMF assessment. But also I just want to mention it will not only be implemented for the DMS assessment, but it will be implemented for ANDAs that self-reference the drug substance synthesis, and it would also be implemented for NDAs that self-reference the drug substances of new APIs.
Thank you for responding to that question. We do have another question for Dr. Raw, and here is the question. With KASA deployment, what are the changes and expectations from the applicant in the submission dossier, including DMF submissions?
Okay, so with the deployment of KASA, in terms of the submissions of the dossier from the applicant, whether it's NDAs or ANDAs or DMFs, there will be no changes to you, because remember, the KASA right now is an internal system that we're doing to enable knowledge management within our system. However, there's a caveat to this because KASA is very intertwined with the PQCMC initiative that Larisa Wu alluded to, and people working very hard in the PQCMC. And once the PQCMC is implemented and there's going to be a guidance that will be issued on this, then it will impact your submission because you would have to have those structures, in the PQCMC. Those structures will pre-populate parts of the KASA. So once the PQCMC guidance comes out in final form, it will impact with your submission. So I think I would encourage you all to. FDA has published some Federal Register notices on the PQCMC. It's on in our website. So I would definitely encourage you to look at these to get better informed about this and engage in the guidance process so that there are no surprises when we implement this.
Thank you for responding to that question. We do have one more question for Dr. Raw, and here is the question. We have heard about the need for SD files for structures in DMF submissions. Given KASA is being implemented for ANDAs and NDAs, should SD files be included in ANDA and NDA submissions?
Yes. So as I mentioned, we are implementing, the KASA for the ANDAs right now for solid orals, and we're going to expand and then eventually to NDAs. And we are capturing the impurities information in a structured format. But and so similarly, we would encourage you, if you do submit ANDAs and NDAs, that you include the SD files in the submission to facilitate our processing of your application under the KASA framework.
Thank you for responding to that group of questions. Moving on to our next panelist, we have some questions that came in for Dr. Larisa Wu. And here's the first question for Dr. Wu. When will the KASA system be available and applicable, and with which IDs will we connect? Will we have one general login for a company?
Yeah, thank you for that question. So I think Andre already alluded to the answer. KASA system is already available internally at FDA and accessible to quality assessors for review of drug product, manufacturing, and biopharmaceutics information that is submitted in the solid oral dosage forms, ANDAs. And as mentioned in the presentations, KASA for drug substance will become available at the beginning of 2023 calendar year. However, it was mentioned already, this is an internal system. And I want to stress that only quality assessors at FDA can access and use this system for their submission evaluation. KASA is actually in a FISPA high security environment, which provides the strictest level of controls to ensure the protection of data. So it is not intended to be accessed by pharma industry. Thank you for this question.
Thank you for responding to that question. We have a couple more questions for Dr. Wu, and here is the next question. When is the ICH M4Q update scheduled?
Thank you. That's a great question. So ICH development of guidance is a relatively long process. I'm actually serving as a rapporteur supporter for the ICH M4Q R2 expert working group. And we are currently in the stage where we brainstorm about the structure of the submission. And we hope that we will be able to reach step one consensus building by the end of next year. Following step one, we would have to be able to sign off the technical document, again, which hopefully it's going to happen at the end of 2023. And that step would be followed by adoption of the draft guideline by regulatory members. Another step deals with regulatory consultation and adoption, and finally adoption of the guideline. Step 5, which would be implementation of guideline, we hope it will happen sometime after 2025. But keep in mind that all these timelines are tentative at this time. They are still subject to change depending on the progress that the expert working group is making at the ICH level. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Wu, and here is the next question. What types of digital tools do you envision sponsors using to submit structured data submissions to FDA?
Again, a great question. And I think I mentioned in my presentation, in order to help us increase our efficiency and effectiveness of our assessments, it would be great if sponsors can submit Form 3938 together with their DMFs. It would be great if you can submit SD files for chemical structures. And eventually, when a standardized form for drug substance synthetic pathway will become available, we encourage you to submit that as well. And just as a note, we are currently working on this standardized form for drug substance synthetic pathway, and we hope this will become available for voluntary use by the DMF holders and the applicants in the future. But I encourage you to listen more information about this in the next session in the presentation called Machine Readable Synthetic Pathways in GSRS and KASA by Tyler Peria. Thank you.
Thank you for responding to that question. We have one more question for Dr. Wu, and here's the question. In the future, will FDA no longer want sponsors to interpret the data in the submission? Will FDA only want?
Yeah, thank you for that question. So the short answer is no, that's not the goal. The sponsor should continue to submit justification and discussions of their data in the applications and DMF submissions. But you are all aware that as quality assessors at FDA, we do perform our own quality risk assessment and evaluation of the data that you submitted. And KASA has been a great IT tool that helps us in that regard. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that came in from Dr. Marlene Kim. And here is the first question for Dr. Kim. What do you mean by V2000 format in your presentation?
Thank you. That is a very good question. V2000 is usually the default file format when you save an SD file in an application such as ChemDraw or other chem informatics software. V3000 is also available, however, it is not preferred. Not all SD file readers may be able to translate V3000 format. So when you save, if it's a default, it'll probably be V2000. But if you have the option to choose between V2000 and V3000, select V2000.
Thank you for responding to that question. We've got another question for Dr. Kim, and here is the question. Can you please explain further how to provide the SDF files in Module 3? Will they need to be embedded in Module 3.3.2.s.3.2 or as a supporting document to 3.2.s.3.2?
That's also a very good question. For questions regarding how to submit an SD file or how to link it or if it should be embedded, please refer to slide 20 for help submitting an SD file, there is a CDER and CBER eSub team that can help you put your application together.
Thank you for responding to that question. We do have one more question for Dr. Kim, and here's the question. Is the use of SD files mandatory in any ECTD US DMF submission, or is it just a recommendation, or is it expected to become mandatory anytime in the near future?
This is also a very good question that we get often. So SD files may be submitted to the ECTD with DMFs and other types of applications such as ANDAs, NDAs, and BLAs. Submitting an SD file with your submission is recommended. It is not required. However, it will aid in an efficient and timely review. If asked to submit by your DMF contact person to include an SD file because you submitted QSAR predictions, then please do so to avoid a delay in the review. And in terms of will it be mandatory in the future, that question I am unable to answer because it will take lots of time to make something required.
Thank you for responding to that group of questions. We do have a couple of questions that came in for Dr. Aaron Skoda. And here is the first question. Is the Form 3938 already applicable for the DMF?
Thank you for that question. The Form 3938 is currently available and can be used right away. In fact, we've been receiving these in most of our DMF submissions currently. It is not currently required, and I do not know the exact date that it will be required, as Dave mentioned earlier. However, we strongly suggest that you submit this form as it's incredibly helpful for us to best evaluate the facilities. Thank you.
Thank you for responding to that question. We have our next question for Dr. Skoda, and here is the question. Are SD files requested for the drug substance itself only, or for all structures, even if it's for the starting materials of the intermediate?
Thank you for that question. So we are requesting SD files for the drug substance, yes, but it is also very helpful to submit the SD files for starting materials, intermediates, and impurities. As Marlene mentioned, it's particularly helpful if we've requested these SD files, but submitting them preemptively is helpful to us, especially as we move into our KASA system, KASA 4.0 in the coming months. So it's not required at this stage, but is immensely helpful and recommended that you submit the structures of all relevant chemicals, including the drug substance, the starting materials, intermediates, and impurities.
Thank you for responding to that question. We have one more question that just came in for Dr. Skoda, and here is the question. How can a DMF holder help with the KASA initiative?
Thank you for that question. So early adoption of the suggested submissions that we've talked about today and we'll be talking about in the upcoming session would be very helpful. So it will help us facilitate the rollout of KASA 4.0 for drug substance review. So for example, as I just mentioned, submitting the 3938 form now will help us to populate the facilities section of our review and make sure not only is it complete, but that it's accurate. This will also help us to avoid significant review delays so that we can avoid having any hidden facilities. Submitting chemical structures in the SD files now, even though it's not required, helps us to submit consult requests to our FDA colleagues and also helps us with populating our KASA 4.0 reviews that will be rolled out very soon. Submitting synthetic pathways when available, as you'll learn about in the upcoming session, will help us also be more efficient in our reviews. And trying to stay up to date on the M4Q process, as Larissa mentioned, will ensure that your DMF is in the currently acceptable format. So all of these things will greatly help us in our review time and will facilitate a smoother transition into the implementation of KASA 4.0 for drug substance assessment. And as reviewers, we really appreciate your efforts on this front to submit these requested files.
Thank you for responding to that group of questions. We just have a couple more minutes, and we do have some questions that came in for Barbara Scott. And here is the first question for Ms. Scott. What section of the ECTD should I place in the SD file?
Hi there, that's a really great question. So for DMFs, the SD file should go into Module 3, specifically in Section 3.2S 3.2, which is the impurities section. For ANDAs, when you create your SD file, that should also go in Module 3, but we would like that to go in 3.2 P 5.5. Thank you.
Thank you for responding to that question. We do have a few more questions for Barbara Scott, and here's the next question. Are there help guides available for the creation of SD files?
I'm so glad that question was asked. We do have help guides available for you, and my colleague will be posting them in the chat box, links to them for your use. So please take a look there. We have one specifically for DMFs and one specifically for ANDAs, NDAs, and BLAs. Thanks.
Thank you for responding to that question. We've got a couple more questions that came in from Ms. Scott, and here is the next question. How do I format an SDA file to be computer readable?
Yeah, another really good question. So when you save the SD file in the Data Warrior free open source software, we want you to select SD file version 2 instead of 3 for maximum compatibility. This ensures that the V2000 version of the MOL files are used, which are the preferred versions for the agency. This is what Marlene was referring to earlier. If you use Data Warrior or any other software, leave the file in the SD file format. It should have a .sdf file extension. And as Marlene already mentioned, for general questions on formatting or submitting SD files, please do reach out to FDA-SRS@fda.hhs.gov. Thanks.
Thank you for responding to that question. We just had two more questions come in for Barbara Scott, so we'll see if we can squeeze these in. And here is the first question. Where can I learn more about SD files and how to format?
Thank you for that question. So I think the best way to answer this is to, I'm going to submit a link again that my colleague will post in the chat box, and you can take a look at that.
Thank you for responding to that question. We have one more question for Barbara Scott, and here's the question. Can SD files be submitted as a standalone submission in ECTD, or are they supposed to be integrated into 3.2 section of the ECTD?
Hi, thanks again for this question. We get this question a lot as well. So for drug master files, again, we recommend the SD file is placed in S3.2 in the ACTD. So this is the impurities module. Thanks.
Well, that's just about all the time we have for questions. We want to give our panelists and our presenters another huge thank you for answering numerous questions that came in.